This is TWIS. This Week in Science, episode number 948, recorded on Wednesday, October 18th, 2023. How do the days keep going? Oh my gosh. Title of the show is, Are Snoozers Losers? I'm Dr. Kiki, and tonight on the show, we are going to fill your heads with maps, dementia, and a snooze button. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. There is a baby crying. My baby. He is sick. Fever, rash, blisters on his tongue. His mother is trying to soothe him. He cannot be appeased. Will not be calm. The discomfort is too great. I would have written a typical disclaimer here, something topical or sciencey, but I couldn't. Thinking is impossible under these conditions. There's a baby crying. My baby. At some point, he will sleep. For how long? Not long. But in the moments in which he sleeps, there will be silence. A blanket of peace thrown over the household. We will step cautiously, avoiding the creaky boards, open drawers and cabinets with delicate care, speak in hushed tones, knowing that there is a baby sleeping. Our baby, but not for long, who upon waking will again be crying. He will get better, be his happy, healthy self again in a few days. But until then, there will be more cries, more tears, and at least one episode of This Week in Science coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki.
Good science to you too, Justin. I feel like I need to be whispering, even though you're hearing me in your headphones.
Yeah, you're fine. You can
After
talk
that
as loud as you
disclaimer.
want.
And good science to everyone who is joining us this week. Thank you so much for joining us for another episode of This Week in Science. We have so much good science ahead to discuss. Woo! I brought stories about mapping the universe, mapping the brain, new old evolution ideas, and fighting cancer with bacteria.
Ooh.
Yeah, what did you bring, Justin?
Let's find out. Oh, I brought that Snoozers Aren't Losers story.
There we go.
I've got an ancient stone tool captured in a painting, but it's not a cave painting. It's 15th century painting. I have Why Lawns Are Evil, and a story that I think is making the rounds, at least about antacids and dementia risk. And it's
Interesting.
yeah, and we're going to talk a little bit about how you even can try to determine causality and something like that.
Right, yeah. Cause versus correlation.
Just a correlation and maybe well, we'll get into it, but it's
We'll get into it.
yeah.
Yay! Okay. I'm excited for your stories. I'm excited to talk about the science. So glad everybody is here. And just a reminder that if you are not yet subscribed and you're here, subscribe to This Week in Science on YouTube, Facebook, Twitch. We are there streaming live weekly, Wednesdays, 8 p.m. Pacific time-ish. And additionally, we are on a lot of podcast platforms. We are podcasters. This is a podcast. It goes, you look for TWIS, This Week in Science. You could just find it. Go on your phone, go trip, 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 and then subscribe. It's so easy. And then, if this is just too much for you, you can go to twis.org. It's our website. We have links to shows, links for subscribing, links for all the stories that we cover. That's where it is. That's where it's at. But
it's
we're
a
here
gateway
now.
website to having all the podcasting apps on your phone devices.
You could go old school and just listen to the show on our website. That works too. Whatever. But, if you subscribe, that's awesome because you get it weekly. So anyway.
Websites are old school. Oh, gosh.
Yeah, I know, right? Oh. It's a really interesting time we're in right now. Yeah. Everything new is old. No, old. Everything old is new. Anyway, time for the show. Let's talk about science.
All right. Yeah.
Yeah. We want to do that. Okay.
Let's do it.
I would like to start with mapping the universe.
That's going to take a while. I think it'll be a very long. And then you got to draw it in 3D, and that's tricky. And then what's the scale? How do you even have a legend on one of those things where it's like, all right, this line represents.
There's so much to be thinking about. I know.
Of 1 million solar systems.
1 million solar systems. Oh my goodness. All right. Well, this last week there were a few efforts in the research world, astrophysics specifically, to map the universe published, and there's some interesting stuff out there. First, I'm going to start with a publication from the Australian National University. Researchers who say in the abstract of their paper that's called All Objects and Some Questions, they've presented an overview of the thermal history of the universe. So in terms of mapping everything, it's more of like two plots against each other of the thermal history and the sequence of objects as they have appeared in the universe. And what they say is that these comprehensive pedagogical plots draw attention to a triangular region forbidden by general relativity and quantum uncertainty.
There's a few of those.
Yeah, but they dive into a place which is the thermal and like what we would call a singularity, the center of a black hole where space, time, mass, all kind of the quantum world loses. Things get weird and wacky there. They say that at this point, there is what they call an instaton. So it's like a new particle, which is their replacement for a singularity. But this instaton, which would be a Planck mass black hole. So basically, like if you were to go out into empty space and you have a black hole that's like the smallest black hole that you could possibly have, that would be at the Planck scale, which is the tiniest thing. This is like getting into string space. When you get into the space where matter is popping into being and out of being, that's the Planck scale. And so this is this place where they're like, if a black hole were to form there, they're going to call it an instaton, not a singularity. But anyway, they ask, do the Planck density and Planck temperature make them good candidates for initial conditions of the universe? They say the plot of All Objects also seems to suggest that the universe is a black hole. However, they map out mathematically how that's unlikely. So it's a very interesting paper that is published in the American Journal of Physics, kind of talking about the mapping of temperature and time and objects in the universe. Like in their mapping, it's fantastic. They've got the log of mass, log of physical radius, and along some of these trajectories, these vertices, they have atoms, bacterium, flea, human, whale, planets, main sequence stars. So there's some very interesting stuff. They go off into this certain areas past black holes that are forbidden by gravity, because what happens inside of a black hole when it's the supermass, we think gravity breaks down at the singularity. There's also a Compton limit in which there's quantum uncertainty that we can't really get past at this point with our technology and our math. But it's I think it's an interesting paper in that it just takes a look at things that we already know.
Yeah.
And maps them out in a way that possibly opens up a space for asking new questions.
Yeah, and one of the interesting things to me too is that whether it is a map of your living room or a map of the entire universe, you still cannot fold it in half more than 7 times. It's a fun bar bet.
It is. Okay. I'm not not an origami person, so but
you can't fold it in half more than 7 times in any piece of paper. Whenever okay, this is so typical of the field of physics, though. I can't ignore the fact that
more coming later with
So first we're gonna create a black hole that can't exist because it's too small, so nothing that was gonna collapse down.
And we're gonna make it a new particle.
And we're gonna create a new particle.
And we're gonna forget about singularities.
We're just gonna ignore it for the
this
moment.
This is a new particle.
And try to build the universe from that starting point. Okay, I guess you had the time to do that.
I did.
And yes, I mean these are the ways that. These theoretical models of spherical cows are the way that discoveries are made, because you're placing these sort of constructed artificially-ish limits on things based on things that are limits that have been observed. And then you see what are the ramifications of this and does it fit with anything?
Right.
So it's a fun field. It's a fun field because it's almost limitless in the ways that you can tear apart the universe and put it back together again. Yeah. Does it do anything? Maybe not, but
but I do like some part of the way that they've looked at things are in these transitions from matter to dark energy after the Big Bang. Like there's the changes in the energy and how things are there. Like they do take aspects of reheating, electron annihilation, things that have happened physically into account. This isn't just making everything up. It's just taking account of everything that we know so far and plotting it. So you can, you can look at it visually and go, oh, that's nice. That's really great. Good job. Yes. Anyway, next story of mapping is this last week we also had the Siena Galaxy Atlas compiled by NSF's Noir Lab telescopes. And this is a digital galaxy atlas for large galaxies. It has just been published in the Astrophysical Journal supplement series. And this Siena Galaxy Atlas is great because it's data from 3 surveys between 2014 and 2017, the DESI legacy surveys, identifying targets for dark energy spectroscopic instruments. Like they're trying to figure out, okay, where's the matter? Where's the dark energy? Where's the matter? What's happening? They used a dark energy camera legacy survey. I mean, we don't know what dark energy is, so I don't know how we have a dark energy camera. But anyway, that's beyond me. They used ground-based telescopes in Chile. We've also got Beijing, Arizona Sky Survey. There's a bunch of other stuff involved. And the science platform has opened this all up to community science and data center. And it has these surveys, 400,000 new objects, galaxies in our cosmic neighborhood that they have taken pictures of. And so this is a total area, optical and infrared, of 20,000 square degrees. Half the night sky. It's one of the largest galaxy surveys ever, and its precise location, shapes, sizes of these galaxies, allowing people to be able to take a look at these large galaxies. It's the preeminent library for this, for people to be able to see what's out there in the night sky and to confirm the things that they are seeing. This is higher resolution, much larger survey than we've ever done before. And so, I mean, I'm just astounded. 400,000 new galaxies to be looking at. And it's freely available for anyone to look at, not just university researchers. It's available for you and you and you and you and you. Anyone can look at it. And it is this.
Where would I go to look at such a thing?
And you can go to sga.legacysurvey.org, where they have the data portal. You can take a look at the bits of the night sky that they have been able to map. You can look at particular galaxies that are in this trove of data. So if you're looking at a part of the night sky, you can see what's there and compare it against what they've looked at. So amateurs and researchers alike, it's a massive trove of data.
I'm gonna need a better telescope.
Yeah,
I don't have one at all, so this is actually pretty easy.
Get your telescopes. And then also in the last couple of weeks, Gaia, which is ESA's mission, recently released a whole bunch of data, 500,000 new stars in a massive cluster, over 380 possible cosmic lenses, and they pinpointed the positions of over 150,000 asteroids in our solar system. Gaia is mapping our galaxy and beyond, but it's also taking a picture of close in. So our solar system is very important. The third release recently contained over 1.8 billion stars of the Milky Way and beyond. And they've got clusters and clusters of, they look like, stars in the sky when you look at them, but really these stars in the sky, it's a star cluster, and that star cluster reveals that there's a whole bunch of stuff going on, and it increases the resolution so that there's 10 times more stars that we're seeing in the cluster core. Additionally, there's 15 times the brightness. So that's how they're seeing all these things. But I'm excited about also the lensing that they've identified, because by observing lensing, what that suggests is that we're able to see back further in time. We're seeing things magnified from back behind objects that are between us and whatever it is.
It's so nice when black holes or gravity clusters can operate like a better telescope way deep out in space. Like, it's pretty handy.
So the old image of Gaia looking at Omega Centauri was fairly dark, and now the center of this image is just fully bright. And there, and they have magnified in on it, they're able to see so many more stars, which means more possible planets, more things out there. It's just I'm so excited about these observations, the mapping that we're doing, to be able to put ourselves in our place in the universe and see what else is out there. And, I don't know. To some people, it might make you feel small, but to me, it just makes me appreciate how wonderful the fact that we're here is.
Yeah. It doesn't make me feel insignificant in any way.
For me,
However,
and
however,
yeah,
there's some awe and wonder there, but I actually I'm so certain that because physics and chemistry seems to be the same everywhere in the universe, that all of the Earth-like planets that are out there are gonna have a form of life. I don't expect
something
that life might be much more than a bacteria, something without mitochondria. Like that might be the kind of high bar for life most places. However, the way it got started here so quickly, and everything that we've learned about how the basic elements of chemistry interact to give the rise of life, all those components, all the organic material that turns out to be so common that we can find it on asteroids floating around in the dark cold of space.
Yep.
Probably all of those planets that are Earth-like, and even going beyond the Earth-like, somewhat un-Earth-like rocky planets will likely have a form of life on them. That's really the exciting thing when I see.
Yeah.
They might not even have to be rocky.
Or icy planets that have methane oceans. There's so many possibilities.
There are more possibilities, but even just limiting it to water and rocky planets, there's a lot of them. Right? There's a lot. And
yep.
And one of the, I know we're sidebarring, but one of the things too is the building blocks for DNA seem to be somewhat naturally occurring, right?
All over, yeah.
The form of DNA, that double helix, is just a structurally sound version of those. So that it's not that it's like, oh, but how would you even have it be the same as on Earth with a double helix? That seems like it would take all this. The double helix prevents things from being pulled apart lengthwise and side to side. It's just more stable. So if anywhere else found that stable medium of these combining elements of chemistry and physics, it should persist. Yep. So I wouldn't even be surprised to find out that DNA as we understand it here on Earth was also a thing. So that life might not even be that different anywhere else.
I swear. Yeah. I think that's the interesting question. Yeah. I don't know. Not that we found life anywhere else yet, but I think it is pretty commonly accepted that based on all of the chemistry, the physics and stuff, if the conditions are right, life will happen. But yeah, I think that's the big question. How different will it be? But
yeah, I've heard there's a way that life could have started here where one of the nucleotides was swapped out for another or something like this, but I don't know. Chemistry being chemistry, I have a feeling it'd be very similar.
We can talk about this a bit later because I have a story about old evolutionary ideas being new again. So yeah, so we're going to come back to this in just a few moments. In the meantime, though, I didn't hit a snooze. Yeah, hit your snooze. No, don't. I didn't hit a snooze button. I just slept through my alarm. You snooze, you lose. What? Is that true?
Okay. So, if you're like me, and let's face it, you are nothing like me, where your hobby of collecting your own nail clippings is limited to your own nail clippings. But if you're a little bit like me, you don't wake up all the way after a first alarm. You hit the snooze button. You drift off for a few more minutes of sweet, sweet sleep. To be all the way like me, you would have to set 3 alarms, spaced out so that all the snoozes that you hit for these set alarms build up to create a crescendo of unending noise.
Finally. Or your child comes and jumps on you. Yeah.
At some point, you just can't figure out how to turn the alarms off of your phone, and you hand the phone to the child. So, so snoozing is a little ridiculous. Everyone who snoozes regularly, the snoozers, the snoozers have to plan ahead for snoozing. If you're a snoozer and you know you're a snoozer, you have to plan ahead. So a non-snoozer has to get out of bed at 11 a.m. So the non-snoozer sets the alarm for 11 a.m. But a snoozer might have to set the alarm for 10:30 a.m., 10:15.
Yes.
Way too early in the morning. And losing considerable time in deep sleep compared to the non-snoozer. So, the debate has been, I don't know where this debate took place, but it exists. Do the snoozers get less restful sleep and suffer consequences from the self-indulgent snoozing behavior?
Right, because snoozing is self-indulgent as opposed to just hearing the alarm and throwing yourself out of bed.
Yeah.
Right?
You're like, oh, that alarm is for common folk, not for me. I deserve more sleep, and I will take it.
I feel like this is, Western culture, capitalist, whatever. But anyway, let's discuss more about these snoozers.
Well, a Swedish study finds the snoozers on average do, of course, sleep slightly shorter and do feel more drowsy in the morning compared to those who never snooze. However, there were no negative effects of snoozing on cortisol release, morning tiredness, mood, or sleep quality through the night, according to the lead author of the paper, Tina Sundelin, researcher at Stockholm University. They looked at 1,732 individuals who answered questions about their morning habits, including how often they hit the snooze button. Many reported snoozing regularly. The snoozers tended to be young adults and evening people. I used to be both of those. Now I'm just the evening people. The most common reason for snoozing? Feeling too tired to get out of bed when the alarm goes off. I completely relate. That's it in a nutshell.
Yeah. Oh God, it's too early. No, snooze. Just 5 more minutes. Just 5 more.
So they did a second study. They invited 31 regular snoozers to spend a couple of nights in a sleep lab, and they measured their sleep in some more detail. One of the mornings, they were allowed to snooze for 30 minutes, and the other, they had to get up right when the alarm went off. So even though sleep was disturbed during the half hour of snoozing, most of them actually did get another 20 minutes of sleep. So that whole, you're sacrificing the amount of time you sleep by doing the snooze routine, not so much. They actually got most of that back. The snoozers also weren't waking up when they did get up from a deep sleep. So that snooze sleep is a much lighter sleep.
Right. So it's more of the REM type sleep. It's not the deep sleep that would be more important, because we know that like a deep sleep cycle takes like 90 minutes. You need like an hour and a half
to go get into it.
To get into it and get out of it. It's like hour and a half like cycles. And when people know they have to get up at certain times, like you go to work, you have a cycle, circadian rhythms are a big thing. Your body kind of wakes up, gets you out of those deep sleeps at a certain time if you're doing it over and over again.
Yeah. I got to the point at one where I had a routine for such a long time that I would wake up like a couple of minutes before my first snooze alarm went off, which was annoying because then I'm like, now I'm awake.
So you can snooze it.
I gotta wait for this thing. And then if I fall back asleep now, it's just going to wake me up in like 5 minutes. Oh, what do I do? It's such a conundrum. So the snoozers performed better on cognitive tests in waking up from the
Goodness.
snooze as opposed to waking up from the deep sleep.
Huh.
There were no other clear effects of snoozing on mood, sleepiness, the amount of cortisol in the saliva. Study shows that a half hour of snoozing does not have negative effects on night sleep or sleep inertia. That's the ability to get up and start the day. The feeling of not quite being alert in the morning wasn't affected. If anything, there were some positive outcomes. When participants were allowed to snooze, they were a bit more quick thinking when they did get up. So, of course, important to remember, a little disclaimer, that this study only included people who are regular snoozers and find it easy to go back asleep after each alarm. Snoozing is most likely not for everyone and should only be attempted by professional snoozers.
Totally a professional here. There we go.
But if you're a snoozer, that's a nice snooze confirming. You can just have that thought the next time the first alarm goes off and you go, oh, I'm doing this for alertness later. This will help me start my day, and then hit this snooze alarm.
Well, I wonder, it's the drowsiness upon waking, right? And if you're snoozing, maybe because you're in that light sleep for just a little bit longer, maybe your brain and all those systems that boot your brain back into alertness and activity, maybe that snoozing gives the brain a little bit of time to actually do that a little bit better. But like you said, maybe it's not for everyone. Some people just need to throw themselves out of bed, and that's the way it works.
This is a good indication that there also could be like some sort of biological difference going on between the snoozers and the non-snoozers. Strategy-wise, if the alarm goes off and you could just get up and you're ready to go, you may not be wired for snoozing.
Right.
And if you're somebody who always, again, I literally have three morning alarms spaced out. Although initially, it was because sometimes in that drowsy, not awake brain, first waking, I would accidentally turn off the alarm as opposed to hitting the snooze button.
Yes.
So to prevent myself from accidentally not hitting the snooze button, I set a second alarm as the snooze button. But now I've gotten pretty automatic at hitting snooze, and so now it is
congratulations,
a constant din of alarms going off at some point.
One thing that I really like seeing here in this data, though, is that the different chronotypes that they've defined, snoozers have the lowest percent of being morning people. Non-snoozers have a higher percentage of definitely morning people. So I think that in itself is this very interesting balance of, how just people are differently wired, and it, how your circadian rhythm works, how everything works together is very important. But yeah, let's get rid of this idea of you snooze, you lose, because it's not necessarily correct.
Yeah.
Unless you're really late for work because you're snoozing all the time, and, it's impacting your ability to do normal things, and you're not maybe you should see somebody for mental health and depression.
I will just point this out because this is not part of the study, but this is always bothered. Yeah, the snoozers, some of us are often late, but we're still wide awake at the end of the day.
Yeah.
Whereas the non-snoozers who woke up 3 hours before work and got there early and were like, on their fourth cup of coffee when they get there, they're checked out by 2:00 in the afternoon. They're like, their brains are done at the end of the day.
It's different.
But you don't get docked at work for, mentally checking out at the end of the day. So anyway, pet peeve.
Just do the work. Get the work done.
But I'm wondering also, evolutionarily, the benefits of this. Like, it may have been, somebody needs to stay up and make sure the raccoons don't run off with the babies, or the bears don't come into camp, or whatever the thing was. And so, like, okay, some of you
I'm
need to stay
imagining,
up.
like baby thief raccoons. Oh my gosh. Okay.
Oh yeah. Oh, of course. Somebody might have been that, like, some of the population community would stay up really late, keeping the fires going, looking out for danger. Other people got to go to bed early and then wake up early and keep the raccoons away in the morning while the
Other people
night
slept.
folk got to sleep in.
Yeah. Different time, and it makes a lot of sense for how a community that is impacted by its surroundings 24/7 would potentially survive the best, is to have different people who are better at being alert at different times. That makes absolute sense. We're going to move on from snoozing to bacteria.
Ooh.
Yes. One of the big issues with treating cancers has been whether it's a blood cancer or a solid tumor. Immunotherapy has been really, really good for blood cancers, but it hasn't been great for solid tumors. And researchers that have been trying to figure out how to use CAR T therapy, which we've talked about before, which are these, modified T cells that go in to specifically attack cancer cells, how to get them to attack solid tumors better. What can they do? It hasn't been really successful in that solid tumor area. And so papers published in Science this last week, researchers used non-pathogenic E. coli to deliver antigens to the tumors, solid tumors, and tag them.
Mm-hmm.
And so there are the antigens on the tumors then that the modified CAR T cells could be attracted to and could grab onto and then destroy solid tumors. They found that when the E. coli probiotic was administered, CAR T cells could be directed to solid tumors. They orchestrated tumor cell killing in mouse models of breast and colon cancer. The system demonstrated cell activation and antigen agnostic cell lysis that was safe, effective in multiple xenograft and syngeneic models of human and mouse cancers. So basically human cancers in mouse bodies. But yeah, so this work is using engineered bacteria to basically go and label and infect tumors with little tags so that our our CAR T immunotherapy can actually work.
Yeah, it's a sort of work that, like, I guess a B cell would normally do.
But it doesn't, because the tumor cells are like, hey, I'm fine. Don't pay attention to me. And that's like, Yeah,
and so here comes this little bacterial creation, and it's and it doesn't register as the thing that's going to tag it. And so it gets away with it, which is fantastic.
Yeah, so this is a new potential for the CAR T immunotherapy, going beyond blood cancers to solid tumor cancers. That would be a very, specific treatment. So it doesn't have lots of the, other problems that come along with chemotherapy and other treatments, drug treatments that kill lots of cells. So the CAR T comes in, is specifically locking onto those cells that have been labeled by the bacteria as tumorigenic and destroys those cells. And so, so it's like the B cells. They selectively attach to the extracellular matrix proteins in the tumor microenvironment They also release a chemokine. So the chemokine is like a signal that's like, hey, CAR T cells, come over here. You want to come over here? This is a good place to be. Come on. Come hang out over here. And then you have Rosie Perez like punching someone in the face, and that's how it works. But
yeah, well, yeah, I can see that. Yeah.
But I was going to say this is potentially going to be. They're looking at it for tumors in what they've looked at mouse models with leukemia, colorectal cancer, and breast cancer. So very much further than just the blood cancers they had previously been looking at.
The whole problem with cancer is that it's a survivor.
Yep.
It's almost like it has gotten off of or it's very much like it's gotten off the track of being part of the body.
Yeah, it's doing its own thing.
And is realizing like, hey, they're gonna get rid of us. That's the little group of cancer cells. If we don't defend ourselves every way possible. And it almost, in a weird way, acts like it's its own organism because it's not listening to the tissues around it. It's not going to pay attention to apoptosis signals or anything like this. So,
but it's interesting. It's like a parasite almost too, because it's like, hey, we're going to
still need the energy
in here, especially solid tumors. It's like, ooh, I want some of that vascularization. Yeah, come on in here.
Oh, I don't like the way you talk. The way you voice cancer is very scary.
Yeah, it should be.
Oh, well, yeah, no, I guess that's it.
Yeah, but I think this is going to be another really interesting tool in the toolkit. And I find it fascinating that as much as we worry about certain bacteria, E. coli, people are like, ooh, my spinach has E. coli and, we have to throw it away, or it didn't get washed well enough. Somebody has an E. coli infection, they go to the hospital. That's a bad thing. There are ways that we can work with bacteria symbiotically, that we can actually engineer them to do things like this that help us survive better. Bacteria are part of our microbiome naturally, so it's not a big step. And this is, I find this kind of stuff really interesting.
And E. coli too has been a mainstay of biotechnology for creating things. So it's used. It's probably in every biotech product that you've ever encountered.
Involved in some level. Yeah.
Yeah. Medical or commercial or whatever. Like, that's a very handy, what you said, tool in the toolkit of biology.
Yeah. Speaking of tools, how about hand axes? I like hand axes. Goodness. I don't know how to use a hand axe. Well, maybe I should learn.
Well, so the hand axe was invented hundreds of thousands of years ago by human ancestors or ancient cousins of some sort. Large stone tools known as the Healin hand axes were used to cut meat and wood. They were used to dig with. They were sort of the Swiss Army knife of Homo erectus. And they were often made from flint. These prehistoric oval and pear-shaped tools, flaked on both sides with sharp pointy ends, have been found throughout history. So we think of, I always tend to think, at least myself, that we've just discovered the ancient world. But the ancient world also discovered the ancient-er world in its time. Though not always with the best understanding of what they had found. So there's some texts from the 15th century of Acheulean hand axes being referred to as thunderstones shot from the clouds. They were thought to be some sort of natural phenomenon. Researchers from Dartmouth, I don't know how to say Dartmouth. Dartmouth?
Dartmouth.
Dartmouth.
Oh.
Say it again.
Dartmouth.
Dartmouth. Okay. They should
Dartmouth. There we go.
They should rewrite how they spell it. Every once in a while, you should update spelling to conform to... Oh, it's probably British, though. University of Cambridge have identified the Moulin diptych, circa 1455, painted by Jean Fouquet, depicts what is likely the earliest artistic representation of an Acheulean hand axe, demonstrating that these objects had an even earlier place in the modern world. And they published this in Cambridge Archaeological Journal. So the Moulin diptych, oh, and there's a good picture of it you got there. You can see you got the guy here there in the red who actually commissioned this work of art. He was the accountant or head of the treasury for the king. And so he had a nice painting of himself next to the taller gentleman there who is supposed to be a saint, who's holding some sort of red and gold book. And on top of it is this very large stone, which seems to be very jagged and sharp and oval shaped, or pear shaped kind of. And actually, now they think this is an Acheulean hand axe that has found its way into this painting. Now, I guess it's Saint Stephen is the one who is the tall one there,
bearing the hand axe,
on top of the book,
yes.
Of whatever book that is. So, and apparently, Saint Stephen was stoned to death for being an early Christian. I don't really know the story, but that's why the stone has been placed in here. Although,
yes,
unlikely, I would say, that he was stoned to death by Homo erectus.
Probably not.
It's still possible. I don't know. I wasn't there.
1455, maybe not. Just saying.
So, but I do love this story for a few reasons. Stephen Kangas, senior lecturer in the Department of Art History at Dartmouth, and the study co-author, had always looked at this painting and thought, that looks like a stone tool to me. That doesn't look like a natural stone. There's something too odd about it.
Look at the edges.
Yeah,
look at its surface.
Edges.
It looks like somebody cracked at it.
And he says, quoting him, it was always sort of struck that in the back of my mind as something that I needed to pursue in the future. And sure enough, the future arrived in 2021. So it's past now. It's now the past. But that was the future once.
And now it is a publication. Okay.
Kangas attended a seminar at Dartmouth about an archaeological site in Tanzania, which has a lot of hand axes. And afterwards talked to the presenter and another professor who he had them look at this painting. And they went, there's a study there. Mm-hmm. And so they studied this stone in this painting. It turns out the artist actually spent a lot of time working on this. They did that thing where they can look below the surface of the paint and they can see the drawing on the canvas before the painting, and they can tell how many times the paint has been reworked, and they could tell that the stone was especially reworked compared to the rest of the painting. The stone object in the painting is similar within 95% to other Acheulean hand axes from the region where the paintings were made. That's important. The researchers counted the flake scars on the surface of the painted stone object.
Wow.
On average, they found 33 flakes on the surface, which was consistent with the average identified on 30 hand axes that were randomly selected from their French hand axe assemblages.
So within a bound of error, it fit.
Yeah. And the stone was painted, had a few, like if you look at that thing too, it's got a little a brown and a little yellow, and there's some reddishness to it, which, I wouldn't have thought a stone tool has, but apparently that color variation is very like the stone Acheulean axes found in the region. They have these discolorations to them. So
Really
it all looks
accurate.
like it fits.
Yeah.
Yeah, it all looks like it fits. And they say about the artist who painted it. Fouquet, I don't know French translation. Fouquet seems to have taken special interest in the stone object, probably because he had seen one that stuck in his attention and imagination. So if that is so, it's then also likely that the artist had never seen a female breast. The other panel of the painting.
We're going to go there, huh?
Yeah, because there's they could count the number of chips that were flinted into this 500-ish or ancient stone tool, this hundreds of thousands year old stone tool. Count the number of the flakes. They could see the coloration, the detail level was so great. But the next panel, there's a woman who appears to have just or is about to breastfeed a child, and there's no nipple. And also the breasts aren't quite in the right place.
They're in her armpits, and
one's in the
pushed off
armpit, one's
to
more
the
centered.
side, and
Absolutely zero detail.
no detail. Because honestly, I bet it was probably blasphemous or looked down upon to have been gazing upon a woman who was maybe he just kind of glanced and was like, okay, I saw enough, and then just sketched it, and I don't know.
And also that painting is supposed to be a virgin and her child, which I don't know how well science was understood in 1455, but I think that would be also unlikely. But it's just the juxtapose between the level of detail in this rock that we can actually now attribute to an ancient stone Acheulean hand axe versus the lack of biological information on the woman in the next panel is just, to me, it says something about the people of 1455.
I think it really does.
Yeah.
I think that is, people can make their own conclusions from there.
Yeah.
Yes. Since 1455, we have moved on. We moved on through years and years. Medieval ages, dark ages, on to the times of Darwin and Newton and scientists that were like, I've got ideas, and they wrote their ideas down in these big writings, and it took a long time for us to really start thinking about how to incorporate all of these ideas together. But now we have the law of evolution, theory of evolution. We have laws of thermodynamics. We have an understanding of natural selection, which was addressed by Darwin. This past week, there were a couple of papers that came out that I find very fascinating because of who's publishing them and what they're talking about. And okay, from my perspective, as a biologist who has studied evolution, ecology, all these things, I'm like, and how is this new? Yet we have a paper published in Nature, and we have a paper published in the Proceedings of the National Academy of Sciences. Both of them by individuals who are mostly chemists, astrobiologists, astrophysicists, and not so much of the biology background.
So either they will have a lack of deep knowledge to assist them in their conclusions, or a complete lack of bias to look at things clearly and cleanly. It could be either way.
Right, exactly. So I've been asking questions of people who are experts in evolution and biology about their thoughts about these papers. And the first paper, which was published on October 4th in Nature, relates to a topic that they present and call assembly theory. And they believe that assembly theory is what explains and quantifies selection and evolution. So both of these papers are trying to take physics and chemistry and bind it together with biology, because similar to general relativity and quantum theory, there's like the math is lacking. There's no binding equations that anybody's really been able to come up with that are like, and this is how it all works. You know, at a certain point, it comes like, it's like, oh, physics is nice and explainable. Oh, yeah, chemistry, that all works. Biology, ooh, that's mushy, you know. And so there's this, even though we can all look at natural selection and genetics, and we can look at genes and chromosomes and DNA and the physics and the chemistry of how those molecules interact and everything. And, you know, so we know like there's stuff happening in there at the basic level. And one of the big tenets of biology, form follows function. So the function, what's needed dictates what the thing looks like, right? Form follows function. This is a tenet. And this is something I've learned over, like, this is ingrained in my mind the same way that ontogeny recapitulates phylogeny is ingrained in my mind. These are statements that anyone who's studied biology is going to be like, oh yeah, I know exactly what you're talking about. But these people are funded in a way that what they're trying to do is reconcile, and this assembly theory paper says that, you know, reconciling biological evolution with the immutable laws of the universe defined by physics. These laws underpin life's origin.
Wait, if they're immutable, how come they're always trying to mute them?
Yes, exactly. So assembly theory, I find interesting. One of the researchers, Lee Cronin, he has been working at the edge of chemistry and kind of this chemical life field for a very long time, like creating molecules that self-assemble, creating molecules that assemble to do certain functions. Like this is the place, and he's the last author on this paper. He's been doing this work for a long time, and he's well versed in all this stuff. But so this assembly theory they're presenting as a framework that doesn't alter the laws of physics, but redefines the concept of an object on which these laws act. So these objects are not point particles, but what they say is that they're entities defined by their possible formation histories. And in saying this, what they're saying is that this allows objects to show evidence of selection within boundaries of individuals or selected units, and that those histories lead to, so basically what an object is because of what's come before, and what an object is now is going to lead to what it's going to become later. That these objects are characterized through forward dynamical processes considering their assembly.
Yeah, but that's not anything new. I mean,
right. So here I am going, everything old is new again. Okay, so this is the first paper that I'm like talking about, and everybody in the evolutionary area is like, oh, okay, I've
we think a horse previously was a four-legged animal that turned into a horse, and in the future, when it's unlikely to be a six-legged animal. Like, I mean,
yeah. The last sentence of their abstract, it discloses a new aspect of physics emerging at the chemical scale, whereby history and causal contingency influence what exists.
But yeah, what's there is going to influence.
Okay, so now I want you to take this in your head. And we're going to move to the next study from the Proceedings of the National Academy of Sciences that was just published just just published October 16th.
Daniel Smith, did they just reinvent time? Yeah, they're like, there are moments where things are a certain way, and there's a before, and there's an after. Yes, that's time. Thank you, Daniel.
And so this new study is called On the Rules of Function and Selection in Evolving Systems. And the media that you might see in the popular stories revolving around this are, these researchers just made a theory of everything. Clickbait. Which makes my alarm signals go ping, ding, 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 because, okay, whatever. And in this study, researchers have, they've basically been like, hey, there are these laws of nature, okay, and we suggest that all evolving systems, including but not limited to life, and maybe this is the thing that separates their thing from all the other things, so they're looking at it from like a everything point of view, are composed of diverse components that can combine into configural states that are then selected for or against based on function. Their big idea is that
so
they
there's
propose
stuff?
There's stuff. Yeah, their big idea is that they propose a law of increasing functional information. And this is kind of interesting. The functional information of a system will evolve, if many different configurations of the system undergo selection for one or more functions.
Okay.
So I was thinking about this in the sense of like, okay, biochemistry. You have chemical interactions. These two things, chemicals, they maybe can do an interaction if the heat gets high enough or the pH changes enough or whatever. But an enzyme comes around that goes, you get to do this interaction for free. And suddenly the molecules do the extra interaction. They are boosted to a different energy state, we'll say. But it's not like a plateau. It's like they went up a hill to the peak and then down into a little valley. But that little valley is like a pond where it's higher than where they started before, but now it's a new pond. So you can have entropy within that pond, but it's not necessarily going to make that entropy go all the way back down to the bottom again. So, I'm looking at this going, though, I learned about this like over 20 years ago in biochemistry, and I don't understand how you think you are making new words here that make new senses.
I think I figured it out.
Can you tell me what's happening?
Yeah. Physics and physicists have nothing to work on right now. They're in that entropy valley you're talking about. It's higher. It's higher than our understanding of physics was 50 years ago. Yeah. But they're waiting for the enzyme.
Right, and they need a catalyst. Them up with
they need that catalyst. They need a heat. Maybe we should turn the heat up on them. Because it does sound like they're now, hey, what do? We could do the spherical cow thing again.
Right. So I feel like there's some stuff happening here. These are big journals who are publishing these papers that are reworking old concepts. Yeah. And so what I'm wondering is it the fact that they're a new reworking that might potentially reframe the way that people think about stuff to allow new questions to be asked? Because, call me naive. Call me like that I don't know what's happening. I don't understand what they're talking about. But I'm honestly at a loss here as to why these are big papers getting big attention in big journals. I don't get it.
It could be like okay. So if I was going to like lead credence to why these would be big papers, without really knowing anything about the scenario, but a good hypothesis would be there's a lot of ground that's been covered in science over the last couple 100 years, 1,000 years, however you want to, however far back you want to go. A lot of ground that's been covered. Maybe there are places where we just learned a thing and have an assumption that this has been documented or has been defined or been well explained by science. Maybe the writers of this paper go, you know what? We know this is taking place, but nobody's written a paper saying it, stating it, putting it into the record.
Right. Maybe it's as simple as that. And that
really could be.
It's possible.
And if they've done so more articulately than has been done in the past, more specifically, more narrowly, without having it be about the evolution of a woodpecker in a specific forest, or maybe that's been done, right, specific to a life form or another system, but never done without reference to a specific scenario. Like, I could see there being some.
Yeah, maybe that's what it is.
And when I hear that, I see this old professor being like, oh, a student being like, oh, I don't know, I gotta do a thesis paper, and so I gotta do a paper on something. Like, well, Sonny, or Mister, whatever, I don't know today, I don't care. Back in my day, we never actually looked into or wrote a paper about just the thing that there's time or that there's stuff. People just assumed it.
Right. And it's
we have two papers, one that says there's time and one that says there's stuff. Now we can move on from there, knowing we have written papers on these.
Yeah. And I imagine as well, there is some amount of, now that we're looking beyond Earth and we're looking into the possibility of the emergence of life from chemical and physical systems potentially different than ours or similar to ours, that maybe this is a place where, because we've only been looking at it here on our own planet, and everybody equates evolution with Darwin, which is not the only part of evolutionary theory. And I just want to tell you, it's one little part.
Early part of our understanding. Yeah.
But it's and I wonder if there's, some aspect of, like you said, physicists going, hey, this is kind of new. And oh my gosh, I didn't learn this, because they didn't take the advanced biology classes that explained evolution beyond just a Punnett square, Yeah, so there's
and to
possibly
clean things up and make it a hard science as opposed to those wishy-washy biologists running around curing diseases while you physicists are still trying to figure out. It's like, well, I spent 10 years studying string theory and that didn't work, and now I spent 20 years looking for a particle that we never found. Oh, I know. Let's just clean up the biological life sciences. Yeah, that's what we'll do. Also, by the way, I will be the not probably the first person to ever say this, but function can follow form. Form can follow function
for sure. It's not one direction necessarily.
It can be. Well, it's like.
But usually. But
when always. You say usually, I don't know.
Because biology is mushy, 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 mushy.
It is. It's like, oh, I've got this ability to see in the dark for this one thing, and now I can use it for all these other things, or whatever. I always think that it's such a two-way street. When change happens over time, as is explained in these papers, new functions can be the new opportunities are there to be exploited by life, and that's where that's why it's so good.
Novelty, right? New functions, novelty is a core aspect of what allows progress or change. Not even progress, just change to take place. But I just found it very interesting that these two studies, kind of on the heels of those mapping studies that I brought up at the beginning of this part of the show, just it's just very interesting. There's, people looking at old things maybe in new ways, maybe putting them together in ways that people have thought about but not put into words in the same way previously. I don't know. It's interesting. New people coming across old ideas for the first time, thinking they're new. Like, I don't know.
Yeah. And to be fair,
nature and PNAS would actually be like, hey, this isn't new. But,
to be fair, though, a lot of research isn't new. Actually, it's a story I'm going to do later. The one on the Alzheimer's, the dementia and antacid connection is an example of something that has been studied and seen a bunch of times. So there's not really a problem with
it's
seeing it again, reporting on it again, just not discovering. Like, we think of science as like, this is the first time anybody's ever even thought of this. But a lot of the times it's, here's another confirmation of a thing in the way things work that we've seen
and
before.
That's
here's
the wonderful thing about science, right? Replication, doing it over again, confirming results, making sure that what we know is what we know, and also looking at old things through new lenses.
I think your problem, Kiki,
I'm old and
cranky. No, is with the science reporting.
I think that's
with the science reporting, telling everybody. This is the first time they've just explained the universe. It's like, no,
people have done this. We've been talking. People have been doing this. It's not new. And like, I know Lee Cronin, he's been working on assembly theory for a very long time. This is not new. And this, come on, everybody. But media wants to do what media wants to do. So,
but if it's the first time you've heard of it,
that's good for you.
Oh, wow. Somebody just explained everything.
Oh my God.
Now I get it all.
Someone just explained the whole universe and everything. Oh my God.
Yeah. It's all sorted out now. Now what do we do?
Now I tell everybody that this is This Week in Science. Thank you so much for joining us. If you were just tuning in, I hope that you're going to stay with us for the rest of the show. And additionally, if you're enjoying the show, please share it with a friend, because that's how we spread the word, is friend to friend to friend to friend to friend, colleague to colleague to colleague. Hopefully you're telling people about TWIS so that more people know that it's okay to talk about this stuff, that we can have fun talking about science and the stories that happen every single week from the lens of information and understanding what we're talking about. Let's go there. Let's go to the source. Okay, if you want to help us grow beyond just telling friends to subscribe, head over to TWIS.org. You can go to our Zazzle link. The Zazzle store is where you can purchase all sorts of TWIS-related items. We've got hats, mugs, towels, great things for you and your friends, pillows you can put on your couch, all sorts of stuff. Some of the proceeds go back to help TWIS keep doing what we're doing. Additionally, if you would just like to support TWIS directly, click on the Patreon link, become a Patreon supporter. Those who support us at $10 a month and more. It's only $10 a month. Oh my gosh. Probably less than Netflix. Oh my gosh. You do that, we thank you by name at the end of the show. $15 and more, you get stickers. $25, I think you get a t-shirt. There's like various levels, but there's all sorts of fun in there. And you get to know that you are a part of making this conversation happen. You're here with us right now. We know you are. Thank you for your support. We can't do it without you. All right, Justin, it's you against the lawn.
The long arm of the lawn. So I asked earlier, and it turns out we don't have a special effects budget or a live band. No. So this is audience participation time. I need everyone to imagine a drum roll in their head. Ready? Go. And the TWIS Award for Best Study Title of the Year so far goes to Hydrobio-Geosociochemical Interactions in the Sustainability of Residential Landscapes. a study on lawns.
What?
Yeah, you can. If you still have the drum roll going, you can stop now. If you can't stop, I'm sorry for putting that idea in your head. The American residential lawn is, for many, an iconic landscape, heavenly in its brilliance. For others, lawns are mean and rueful of the Western dream. About half of homeowners in the U.S. use fertilizer to keep their yards green and lush. Some proportion of the nitrogen in this fertilizer then works its way into the broader environment and can have negative consequences, including algae blooms and deoxygenated waters, depending on where those waters end up. So Peter Grofman and colleagues studied residential landscapes in the Baltimore, Maryland metropolitan area, which drains to the Chesapeake Bay, which is already not what you would call pristine. Seeking to identify locations, hot spots or times, hot moments, with disproportionately high rates of nitrogen export. The authors went to lawns in exurban, suburban, college campus settings to measure nitrogen export during simulated rainfall events. The authors also used household survey data collected from the Baltimore Ecosystem Study, long-term ecological research that has been going on for almost 20 years. We're at about 20 years. So they also did their own survey of 3,836 Baltimore area households. I thought it was pretty interesting what they ended up finding here. So, although all of the export hotspots were found on fertilized rather than unfertilized lawns, the survey data, 48% of the people surveyed incorrectly believed that they did not live in a watershed. More than 60% of those surveyed did not know if nitrogen negatively affected area waterways. Bummer fact, it does.
Yeah, it does.
And what they then found was that support for policies that would limit fertilizer use among those who were using fertilizer was broadly high. Some types of restrictions even garnering support among those who fertilize, who love their lawns. So about half of households signaled that they would be interested in converting lawns to features that reduce nitrogen export, rain gardens, if the conversions were subsidized and made as easy as possible. So the idea is somebody's got a house with a lawn. They like the lawn. Would they rather, would they be cool with having a garden that didn't need to be fertilized and didn't need as much water? Yeah. They just don't want to they don't have time and money to tear up a lawn and build a whole thing.
Why do you have to tear up a lawn? Just let the weeds take over and then mow those.
Yeah, some people do have I don't want to call them people superficial, but keeping up appearances and stuff, whatever.
What?
You don't want to do that. You can say that because you live in a place that rains, 250 days out of the year. So even you're going to have nice green, lush looking weeds.
But except for the moles. Then the moles come through and they have the holes and the moles. Oh my gosh.
Still talking? You were talking about moles last week too. These moles really got to you.
They really, I don't understand how anyone has a nice lawn who does not invest in poison and traps and the bad things, because these moles, they're just there. They do their thing. Anyway.
You just need to get pet ferrets. That's all.
Oh.
Are they legal? I don't know.
I should check. Not in California. Maybe in Oregon. I don't know. Okay. Anyway, on with the lawns or off.
So the main conclusion of this study really is just that that change is possible and even desired. People just didn't know their role in things. The researchers were actually kind of taken aback by the fact that how willing people seemed in these surveys to make the changes. They just weren't aware that they were part of a problem. So, yeah, people needing to know their role in the larger ecosystem, what the trade-offs are, and a little incentive could go a long way. So, sort of think of it as if you're a city who's contemplating a multi-million-dollar cleanup of your bay. And, what do? We can change the chemistry. We can have these machines.
Yeah.
We can have a wastewater project. We could do a thing. Or part of that, you could just offer an incentive to redo people's lawns, like a mobile lawn transformation part of the city. What are those tree cutting people? Arborists? Hey,
Arborists.
The landscapers for the city, they can't be that busy, especially not in Baltimore. There's not that much open green land there, Put some people to work transforming lawns into these water gardens or whatever they're calling it. Anyway, sounds like there could be a win-win there.
Yeah, so you have municipal level policies that go to the HOAs and supersede whatever HOAs say. So then it's like, all right, now everybody has to do this kind of stuff. And I really do think that there is a place there. There was another study that came out this week related to more climate level adaptations, and discussing how the majority of adaptations that are taking place are at the individual level. Like, people are doing stuff, but really, like, it's the government that has to do, like, there's a gap between where the government is talking and making regulations and then actually having that passed down through the level. There's like a gap there. And this is one of those places where people are used to the normal, used to we're supposed to have a lawn because that's what you do. They don't know that lawns can not be great for the environment. They don't understand what's going on there.
I don't know what this is, but in the chat room, Harjar is saying, look into a vibrating mole repeller. Works for us real nice.
Look, I have
vibrating mole repeller?
I have not gotten into the vibrating mole repeller because
do you know what that is? Have you heard of this?
The tremors worm to come and eat my child. So, I mean, Kevin Bacon doesn't live near me at all. So, or Ed, what's his name? Who was the other? Yeah, I don't have.
I gotta go watch Tremors again.
You do. It's like actually several movie series that is fantastic. And it just gets better.
Is it several? I think I might have seen that first one.
The B movie aspect of it is just...
It's really well done.
Yeah, chef's kiss. Oh my goodness. Yeah, take us off from lawns to somewhere else, Justin.
Oh yeah, it's I'm up again. Here we go. This is the last one. This is research at Copenhagen University Hospital and Aarhus University, Denmark. Looked into the potential association between protein pump inhibitors, which is the antacids that are used to suppress stomach acid production, and they found increased dementia risk. This is a pretty good sized study. This is a nationwide Danish cohort of nearly 2 million individuals aged 60 to 75, done over 2 decades. And it included individuals without a prior dementia diagnosis or treatment before being part of the study. So the study got narrowed down to 99,000 individuals, 99,384 individuals who developed dementia during the study, and another 469,000 who were used as a control group. The antacid use was associated with an increased risk of all causes of dementia. Particularly, they found in the group between 60 and 69 years old. As people got older, there was less of a correlation. Once you get to 90 years old, there was no real correlation that they could find between dementia and antacid use. But it was very significant for the younger groups. And what's sort of interesting about this is there was like the longer you'd used it, the higher the chance of having dementia. But it was also any use. So even if you used it for, say, 3 months, it indicated an increased risk of dementia. So I saw this and I'm like, does not, to me, look like it could be causal. Like there's that possibility. That's a possibility. But it almost looks like an early symptom of dementias.
Yeah.
Will be effects on
Digestion.
the gut and increased acid. And so there have been also previous studies that have shown there are, in Alzheimer's patients, there is a drastic change, a really identifiable change to gut microbiota.
Mm-hmm.
There was a Parkinson's study that also showed that there was early indications of changes in the gut microbiota and so likely what's happening is, so there's the enteric nervous system. That's the nervous system that goes from your throat down through all your stomach and your intestines.
There's also
and it's neuronal
the gut
cells.
Brain connection. There's got the vagus nerve that's involved.
Yeah.
There's, serotonin, and, there's so much interconnected there. Yeah.
So, and the authors also point out in their study that further research is needed to explore the mechanisms underlying the association, whether it varies with dementia types, what is the causal or reverse causal, possibly. But it looks very much like that's based, the way I'm seeing it is it looks very much like that increased stomach acid might be an early clue of dementia risk. Which, if that's what they end up finding from this, that it's in itself could be huge. Because for some of these individuals, the use is 10 years, 15 years of this.
And for some, it's not something that's necessarily prescribed, because you can take antacids just over the counter. You go and get yourself whatever.
Which is actually partly why I think it's a little helpful, the Danish cohort study. Because with the universal healthcare, if your doctor recommends it, you're likely to have it prescribed and paid for, as opposed to needing to buy everything over the counter, or like sort of. I mean, you still can, but why would you if it's covered under medical? Because that's one of the hard things in these studies is it at a certain point, unless you go back far enough. 90s, I guess, you still had to be prescribed. It was at some point there in America became over-the-counter versions.
Yeah.
But initially there wasn't. So the problem then is any studies done after that can't find it in just the large data sets, because it has to be
only the prescribed
self-reported
stuff.
kind of a thing.
Or self-reported,
Yeah. But I found this interesting because, even if somebody's been using it for a long time, that might then indicate a slow growing form. But because there were so many that were in the only used it for 3 months or 4 months or a year or something like this, that then had this bump in dementia use. On the other hand, because of that enteric gut-brain connection, is it possible that antacids are in there disrupting normal microbiota preservative functions? Are they
Yes.
Right. Are they interacting with disrupting the lining of the stomach or the
they're changing
are they making
the
changes
pH.
that could be causal?
Yeah,
so it could be.
A lot of them are calcium or magnesium related, right? So they're non-prescription are milk of magnesia, kind of stuff, or calcium citrate. Or there's the stuff that's going in there is
making
binding
changes.
up hydrogen and making changes, and is adjusting the acidity of the gut. Why is the gut that acid in the first place? Because the bacteria that are probably there and what's happening in their metabolisms and what they're producing. So is it cause, is it correlation? That is like for sure something that needs to be determined. But it's
So, and it does look, according to this, that it is correlation,
yeah,
but could also be causal.
and it
but
could
the
be
reverse
both,
causal,
depending. It could be both
and it
for
could
different
be
people.
the way they work together.
Yeah, synergistically, right?
Mm-hmm. It could be accelerating cases. It could be decelerating cases. But the fun thing is, if in case you see this, antacids cause dementia, it might not be that clear. It might not be that straightforward. But it is really an interesting intersection when you have something that is a real strong correlation find.
Yeah.
But could be multiple causals still.
I find that the dementia occurring after 90 years, it's like if you're making it to 90 years old and
you're
then
fine.
you take antacids, it's like you're fine.
Yeah.
Everything's great. It's like you're doing just great. It's good.
and that it's also part of why I got the feeling that the direction works from Alzheimer's before use, at least in this cohort that they've got here, because if you hit 90 without having to address your stomach, then that's when the dementia hits. It's like, I don't know. It seems to fit better if dementia is causing the stomach acid, which, like you say, could be from alterations in the gut microbiota, which could be from the gut-brain connection. But again, we don't know which way things start. The other study I was referencing that was from a while ago almost made it seem like Parkinson's disease starts in the gut. Like they made kind of an argument that this was a gut first, and
I remember
then
that.
Like
I remember that a while back. Yeah.
So
hasn't been confirmed yet, but yes.
No. I mean, but the fact that that's even a path, right? The fact that a lot of these correlations and connections lead back to the gut. We really need to understand the effects of the gut on the brain, because it could be the source of the disease, or it could just be an indicator of what's going on in the brain. It could be trying to tell us, gurgle, gurgle, gurgle. Hey, I got a problem up here, and here's how I'm going to talk to you by using the stomach.
Yeah. Well, thinking about the brain, researchers at the Salk Institute and a large number of other organizations around the world, the U.S. National Institute of Health's Brain Research through Advancing Innovative Neurotechnologies Initiative Cell Census Network, known as the BICN, BICCN, hundreds of scientists working together, also with Australian researchers, European researchers, like people everywhere, all over the place. They've been working to map the human brain. So they've been mapping fly brains, they've been mapping mouse brains, they've been doing all sorts of mapping. And then there were like these this kind of is like cell types in the human brain, and we've got some little things going on with the human brain, and like this is really awesome. And then they kind of were like, these techniques that we are using to identify subtypes of cells in mouse brains, we're gonna do it in humans. And so this is like a decade almost in the making, these studies. It was like a set of 21 papers that came out to publish all this stuff. But this brain initiative, I love in this Salk Institute PR press release, they have the word, they're describing the full plethora of cells. I think it's honestly a writer's goal to use the word plethora.
To be honest,
possible.
It's one you try to avoid because it can be used up. But every once in a while, you're like, oh man,
of cells.
Plethora is the only word that I could use to describe all of these things.
Yes. Well, this menagerie of cells that they have described, They have revealed more than 3,000 different cell types. Lots of them are new to scientists and to science. And the way that they are new is not just in what they look like their form, but also their genetics and how they work together. So looking at similar genes that are expressed, one cell from the back of the brain might express or have a gene expressed with an on switch, whereas in the front of the brain, that same gene gets expressed by an off switch. So the same, cell types and the genetic differences between them are making up what these researchers are using and saying is this atlas that's going to give a much better understanding of the human brain and how different cell types work together. So they're looking at developmental histories, different functions, the mix of neurons and other cell types across different regions. They looked at the brainstem, which is not as studied as other areas in the brain. They found a lot of neuron types that had not been described before in the brainstem, which is the connection between the brain and the spinal cord. Pretty important area, right? You'd think. They use chemical markers that switch genes on or off in more than 500,000 different cells. They found that molecules acted as switches to identify about 200 different brain cell types. They also have genes for how, or a different understanding now how different genes might contribute to disease risk. So like we're talking about Alzheimer's, Parkinson's disease, other neuropsychiatric disorders, bipolar disorder, depression, schizophrenia. They're looking at how these different genetic switches are able to influence gene regulation and risk of neurological diseases, and not just in neurons, but in microglia as well. Microglia are some of those support cells which are involved in cleaning up dead, damaged cells. They're also involved in some local cell signaling. They are very important for basically helping to manage neurons. They keep the neurons in line. And so microglia and understanding how they're working with the neurons themselves is a big aspect. But it's a huge, huge undertaking. Like I said, almost a decade in the work that they've been doing. And they've gotten to this point now where they have all sorts of new cells, 3,000 new cell type, new independent, individual types of cells that they can start looking at. So more questions can be asked.
And it's unfortunate that they wasted all that time when physicists, and I think a couple of afternoons, wrote 2 papers.
Oh, yeah.
One that said that time is the thing, and the other said that stuff exists, which is all that we really needed to know.
Oh, and slap. Yeah. So it's very exciting. Who knows where it's going to go from here? There's this whole package. If you're interested in brain stuff, you can go find it. Anyway.
Where's the gut? Where's the gut brain?
It wasn't included in. So they're just looking at the brain itself. They got to the brain stem, right?
Right, they got to the brain stem, but those are neuronal cells embedded in from the esophagus through the intestines. That's neuronal cells. That's brain. You have a brain in your stomach. You have a brain in your gut.
Yeah, and so one of the very interesting things moving forward from here is maybe they can start to address some of those questions about that gut-brain connection with or without the microbes and how they're involved, and how the vagal nerve innervates and influences the enteric nervous system. And they've gotten to a point now where basically they're genetically barcoding these nerve types, these cell types in the brain. And so this barcoding is going to lead to an ability to compare against other cells, other things that they look at. So maybe the, enteric nervous system is big also. Maybe that's the next step.
So it is. And so here's how I when you're talking about form following function, all these sorts of things, here's one way to think of it. The brain is connected very directly to some senses, the audio system, the visual system.
Oh, the senses.
We have nerves running through our body that allows us this touch system.
Proprioception.
Before we had a lot of that going on, the way that the body, the brain, sensed the environment was through the gut. What did I just eat? Is that a good thing?
Or bad
Is that
thing?
not a good thing? This is what I know of the world. This is how I sense the world. This is how I know the world exists. It's in my stomach. To an extent, right? They had these, very primitive forms of, there's a thing, I can see it or can hear it or can feel it, sense it then I eat it. But now I have to figure out what it is.
Yeah, so I like to think about that also. I mentioned earlier, ontogeny recapitulates phylogeny. It's so the development of any mammalian organism, humans included, it starts with that blastocyst, and what happens immediately? It forms a tube through the middle, and that is the gut tube.
First
and that's
sense.
where it all.
Brain.
It's the first thing that starts. From there, everything else goes.
It's stomach first. We always think, the brain, and then there's all this stuff got attached. No, stomach first.
Gut first.
So maybe we've been ignoring the biggest sensory organ, the information back and forth. Maybe the brain that's really running things.
It's not as
isn't
exciting.
The one on your shoulders.
It's like your gut. It's the thing that, it makes you feel bad. It's like embarrassing sometimes.
It's
like,
not that. It's just trying to talk to you. It's just trying to tell you things.
My final study published in the Proceedings of the National Academy of Sciences this last week. Researchers at Kyushu University have taken brain immune cells and turned them into neurons and treated stroke injury in mice.
Hmm.
Yeah. So they took microglia, which, like I mentioned, they're normally for getting rid of dead or damaged cells. They go to the site of injury, they replicate, they're like, we're going to take out the garbage. Ta-da! And so these researchers were like, hey, these microglia are just in the right place and they're dividing. They're doing this cool stuff. And so they gave mice strokes. And then afterwards, they used a viral vector, a lentivirus, to put DNA into the microglia cells that made them turn into neurons. It was a DNA for producing what's called neuro-D1. It's a protein that leads to the rest of the conversion into a neuron. And so these neurons that had been infected with the lentivirus, they turned into neurons. 8 weeks after, they integrated with the rest of the brain, and these mice that had reduced motor function and deficits related to the stroke saw improvements. They were able to move better. They were actually improving faster than mice that did not have the same treatment. So, not taking cells from somewhere else in the body, not taking cells from some other animal, not doing just minorly shifting the form and function of the cells that are already in place within a site of injury in the brain led to help.
Yes.
It's interesting. So anyway, yeah, we can help stroke in mice. We'll see if we can do that in humans at some point in the future.
We did. I love that we did the brain atlas on mice first.
Yeah.
So, again,
thanks.
Someday in the post-human world,
when the mice run everything,
when the mice rise up, become sentient, they will find in the archives and the records the cure to every mouse disease left to them by the ancients, the giants, the ape, the hairless apes that wandered the earth making the world, preparing the future world of Mousedom so that it could be disease-free.
Disease-free,
I hope they
mouse
appreciate
kind. At least we can set the environment to help some organisms out there in some way. Rachel had a stupid question. Why don't we have teleportation, Justin?
Is that stupid? I don't know.
think it's a stupid question, actually. She says it would make so many lives so much easier, and I think we as a society deserve that. What's the legit sciencey reason we can't teleport yet?
Well, because you still have to go everywhere.
Yeah,
You still
have
combine.
to put all the pieces back together.
Look.
Or you have to figure out what they were in one place and then
If we're talking Star Trek, if you're talking Star Trek, here's the thing that is a problem with the teleporter in Star Trek. They are murder devices.
Yes, they murder the person in the place and they recreate them in the next place. That's
You could
not
just as easily go into the transponder or the teleporter and have another version of you pop up on the planet surface without killing yourself. There would be two of you. So, and that one wanted to come back. You can't actually transport them back.
And then you have to kill the alternate version.
You would have to destroy them down on that planet and then create a new one up. And what would be the point there? So you would just really, what you want to do is have a clone of yourself out there in the world doing stuff and then reporting back and then.
No, because then they're not you. They're someone else. They're not you. They're a different. They're a different person. That just is.
By the time you can deconstruct a human and teleport them, you don't need to destroy the first version. You could just be cloning and making a second version.
Right.
That's the problem with it. And then, because the real problem is you only have one toothbrush. So you transport, and then the other you comes back, and now you're like, I gotta share your toothbrush with this other person.
Becomes a shiv, and that's the murder device, not the teleporter.
Then they go and teleport somewhere, and then there's three of you, and just in a matter of a couple of episodes of Star Trek. That ship is overflowing with Kirks and Spocks and Boneses, and they're all like, I'm the captain, I'm the science officer. Oh, well, so am I. Yeah, so are we. Well, I'm not a doctor, but I am. This is nonsense.
Science, it's complicated, but the legit sciencey reason is that we can teleport little atoms, and they're happy. It's that quantum butterfly entanglement. That's what we're doing. It's not no, we're not doing it yet. We're not there yet.
And really there is no that whole thing with the quantum parts. If one is up, one is down. Here's two letters hidden in a secret package. One says up, the other says down. That we know. I can put this one on the moon and open this one, and now I know what the other one is. But it doesn't mean that I have transmitted any information over from here to the moon. It just means that if this was up, the other one's going to be down.
Yes.
That's all it means. We haven't done it yet. We haven't really teleported it.
No teleportation. It's entanglement, not teleportation. Yeah.
And entangled just means the information is entangled, which means so it's like if you put an up in one envelope and a down in the other envelope, that information is now entangled. There you go, quantum physics, and
oh my
it's
gosh,
a little
we're
more
having
complicated.
A conversation. We're so entangled, Justin. Oh my gosh. Let's finish this entanglement. Have we come to the end of the show?
Oh yeah. Is this the end?
Yes.
I'm trying to think. Do we have more? No, there's no more stories.
No more stories. We've come to the end of the show. And I would love to thank everyone who's been in the chat room, in the Discord, chatting through the whole show. Thank you for your thoughts and your ideas and everything throughout. I know that some of you are here and don't make any comments. Thank you for being here all the same. Anyone who's here live, thank you to Fada. He's off doing an improv showcase tonight. So
Yeah, break a leg.
yeah, but he's still going to be doing our show notes and social media. So thanks to that. Identity 4, thank you for recording the show. Gord, Arnlor, others who help, and everybody really, for being nice in the chat rooms. Let's all be good people, okay? Let's all be nice to each other. And, of course, Rachel, who helps edit the show, and our TWIS patrons. Thank you so much for all that you do to help keep the show going. Thank you to Arthur Kepler, Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Bob Coles, Kent Northcote, Rick Loveman, George Corris, Pierre Velazarb, John Ratnaswamy, Carl Kornfeld, Chris Wozniak, Vegard Shefstad, Donathan Stiles, a.k.a. Don Stylo, Ali Coffin, Reagan, Shu, Brew, Sarah Forfar, Don Mundus, P.I.G., Stephen Albrand, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredus 104, Sky Luke, Paul Ronovich, Kevin Bearden, Noodles, Jack, Brian Carrington, David E. Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Dan, Christopher Rappin, Richard, Brendan Minish, Johnny Gridley, Jimmy Dave, G. Burton Lattimore, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Ronnie Lewis, Paul Rick Ramus, Philip Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O. Adam Mishkan, Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Baldy Disney, David Simmerly, Patrick Pecoraro, and Tony Steele. Thank you for all of your support on Patreon. And if you want me to read your name on this list, head over to twist.org, click on the Patreon link, and support us at $10 or more per month. On next week's show,
We will be back Wednesday at 8 p.m. Pacific Time or Thursday, 5 a.m. Central European Time.
which is the same
Live from our
time.
YouTube and Facebook channels and from twist.org/live.
And if you want to listen to us as a podcast, just look for This Week in Science wherever podcasts are found. And if you enjoyed the show, make sure to get your friends to subscribe too.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twist.org. And you can also sign up for a newsletter, although why would you bother? You're getting so much content here.
You want the newsletter? That'd be great. But you can also contact us directly. Email me, Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, Blair at blairbaz@twist.org. Make sure you put TWIS into the subject line so your email doesn't get spam filtered into some kind of, I don't know, everybody's gonna make old things new again paper that makes me upset that I won't read.
I have also heard that we are available for contacting at a social media site
At
that
a
has
social media. Yeah.
that does not seem to have a name, or used to be called Twitter, where we are at Twist Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you tonight, please let us know.
We will be back here again next week, and we hope you'll join us again for more science news.
And if you've learned anything from the show, remember,
It's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week This week in science. This week in science. This week in science. This week in science. This week in science.